activity in coastal environments can have a significant benthic component. In shallow coastal regions, light can reach the sediment surface, resulting in growth of benthic algae such as mat-forming diatoms. Since light is the limiting factor based on water depth in tidal areas, understanding the photosynthetic downwelling irradiance, which is the PE relationship, is necessary for quantifying benthic primary productivity on a day-night cycle. Over here in my photo, I have a picture of basically what bare sediment in the estuary looks like, which is the top one, and then what a mat-covered sediment would look like in the estuary. It's hard to tell the difference because when diatoms form mats, it's still like a brown sediment. It's just like almost like jelly, and that's what the mat would look like under a microscope as well. And each little diatom is one little frustule. A summary of my study is that I'm going to determine the rate at which benthic macroalgae with a focus on diatoms contributes to ecosystem primary productivity across a transect in a mid-coast Maine estuary. And to do this, there are two different components to understanding the dynamics of oxygen within the transect. The first one is a total oxygen flux out of the sediment, which is done by quantifying the rate at which oxygen is produced and mixed into the water column by recording the oxygen increase at a well-defined light level for 1 hour to determine the total oxygen uptake rate out of the sediment, which is the TOU rate. And then another way to do this is the oxygen dynamics within the sediment. And this is done by quantifying sediment O2 concentration within the sediment with depth at the same well-defined light levels for an hour to determine the diffusive oxygen uptake rate, which is the DOU rate. My study site is a transect across mid-coast Maine estuary in the upper part of it called the Damariscotta River Estuary, and I refer to it as the DRE. It is an estuary that is quite long, stretching like 30 kilometers. It also has an output into the Gulf of Maine, and it is also connected to a freshwater lake, so it has freshwater input and tidal mixing every day on a tidal cycle, and it's very dynamic and nutrient rich. It is the host of a large aquaculture industry for the state of Maine, and also is the location of the University of Maine's Darling Marine Center, which is their marine research lab, which is where I did my research out of. And it is a highly researched estuary, but has had little benthic research into it. The photic zone reaches all the way down to the sediment for most of the DRE, and there is a phytoplankton bloom both in the spring and the fall. Across the transect, the water ranges from about 1 meter near the shore to about 8 meters in the middle, so it's not that deep. And the dominant form of benthic algae, or benthic diatoms in the area of the transect is the pennate diatom gyrosigma, which is known to migrate vertically within the sediment based on, they think, light, like attenuation and light availability. So at each site across my transect, during the summer of 2019, between the months of June and August, I collected 6 long cores and 6 short sediment cores, and I would determine which ones were good just based on similarities and lack of disturbance during the sampling process. And I would transport these back to an environmental chamber within a lab where the temperature of the chamber would be set to the bottom water temperature during sampling time, and they would be contained in large tanks with UV filtered estuary water, and then they would be left overnight to minimize disturbance from the sampling while being aerated. And then to get oxygen levels between the 2 different experiments, they were both done simultaneously at the same time, and the total oxygen incubation was done in the large black tank, and it was started based on the cores would be capped in the dark to start off, basically minimizing any oxygen bubbles that were in the top of the caps. And once they were all capped, as well as blank core to minimize any basically production consumption happening already within the water column. And it would start the oxygen would be start measuring once the caps were put on and the mixing of the overlying water column was started, and it was measured every 5 minutes for 1 hour light intervals ranging from 0 to 120 micromole photons per meter second. And this lasted for an 8-hour interval incubation. And once that was completed, the cores were sectioned for the diatom mats, and also 1 centimeter below that for cell counts. And at the same time, after each light interval in a smaller tank, oxygen microprofiles were done in a smaller core in 3 different spots after each interval to measure oxygen concentrations within the sediment after the same light intervals. My data analysis consists of the photosynthetic irradiance curves being fit to growth parameters to determine the total oxygen uptake and DOU rates for Pmax, alpha, and respiration. Cell concentration plots were also done from the diatom cell counts from each core. And then there will also be light and tidal measurements for the in situ light surface and water depths recorded daily by the University of Maine. Has a buoy that records right across from my transect every day during the summer months. And then I will determine the light attenuation coefficient based on water column profiles that were also taken during the summer of 2019. And then therefore, for each day I sampled, I can calculate the bottom water light based on the surface light and then my attenuation coefficient. And then from here, benthic primary productivity estimates will be calculated using the parameters from the PE curves, and then the surface irradiance, attenuation coefficient, and water depth. So first, for the total oxygen uptake incubation fluxes, plot A is the raw oxygen data with the light intervals in black, and then each core that was from the transect site. And then from here, the rate of oxygen uptake, in the overlying water was calculated for each core and then plotted in a P versus E relationship, which is the rate of oxygen production in millimoles per meter squared per day with the light level, and then was fitted to equation 1, which is the growth curve, to determine the Pmax, alpha, and respiration parameters. And as you can see, most of the plots on here are very similar, except for the middle column, which seemed to be significantly lower for Pmax and leveled off quite quickly compared to the other sites. And then next, the dissolved oxygen uptake rate was done. And this was done by the raw oxygen profiles were plotted, which is A, which you can see the intervals from 0 to 221 micromole photons, where it starts off decreasing oxygen at the lower light levels and dark until around 40 microns to 60, where you slowly start to see a subsurface oxygen maximum zone where the oxygen is decreasing actually within the sediment. And then the depth of the oxygen zone also increases as it is diffusing both up, like out of the sediment, and also down into it. And then from here, the flux out of the sediment, the diffusive flux out of the sediment was determined based on the Berg method, which is done by using equation 2 to determine the least number of zones of production and consumption needed to balance the diffusive uptake equation, which is the equation number 2 down below, which then equals the net oxygen flux out of the sediment. And plot B is what it is at 0. So red is all the basically consumption, and blue boxes are production rates. So basically when it's dark, like there's only consumption, and then as you move along to 40 microns where we have that subsurface oxygen zone starting to produce, we have production. And then once we get to the higher light levels, we have way more production than consumption. So therefore it's a net productivity when you get to the higher light levels. And then from here, after each profile was done, then I got a rate the same as the rate from the total oxygen uptake. And these were done the same way, where the P versus E curve was fitted using equation 1 to determine the parameters of Pmax, alpha, and R. And for these ones, we didn't know during the time, but Pmax wasn't being reached in the sediment. So basically, it makes sense now that not enough light was actually getting down in compared to what the surface was for the total was seeing. So Pmax wasn't reached, and then since it's not only fitting for the alpha during these things, we decided to do a linear fit just to make sure that we were getting the alpha parameter done better, because that is actually the point where our in situ light levels are reaching, is in the alpha range of the curve. So then these curves were also fitted on plot B for a linear fit. And for these sites, they were all very similar. And then now I have all of my data compiled into one chart here. And for the total ANOVA tests were done on both the total oxygen uptake and the diffusive oxygen uptake. And then it was also done to compare the two. But first, to looking at just within the total oxygen uptake rates, across the transect, the data was statistically similar, except for the middle column site, which seemed to be lower. And then after running the test, it was statistically lower. So then, based on these parameters, it was decided to use the average across the entire transect, except for the middle site, to make determining the rate easier. And even looking at the image over here on the corner of the cores at the end of each experiment, the middle site just has like significant less like diatom coverage on it, even after like being exposed to high light. And then for the diffusive oxygen uptake, we didn't run an ANOVA test on the Pmax because Pmax wasn't reached for some of the sites. And then for alpha for it was statistically similar, except for the shallow sites differed. So they seem to be statistically higher than the rest of the column. And then to look into why certain sites are producing more than others, the cell concentration was counted and then plotted against the parameters determined for the TOU. And the Pmax, it has a linear trend to it, and this infers that to the difference in the rates at each site due to different cell specific increase compared to the middle of the channel. So statistically different for Pmax due to the decrease of biomass present for the middle site. And then for alpha and respiration, the plot seemed to be more flat, so it didn't infer that. And this could be different due to basically higher chlorophyll present in the cells there, instead of just a greater biomass present compared to the other sites. Comparing both methods, an ANOVA test was also run to compare the two, and it showed that the site West Deep, which is WD, was statistically different for alpha compared to the rest. And looking at these plots, you can tell this just based on the trends they follow, because for every other of the plots, they seem to follow the same trend during the linear portion and then differ just slightly, just being lower. Whereas for West Deep, they don't follow the same track, and it is significantly lower in the linear portion of the plot as well. The conclusions from this is that the in situ light range from 6 to 100 micromole photons is within the linear portion of the PE curves, and alpha is the main concern then. based on the TUU in the middle of the channel site, the transect has lower productivity than the other sites, which is also what was kind of expected because it receives the least amount of light based on the in situ natural light range, and it is lower, basically ranging around like 10 to 20 micromole photons, even at like midday at the highest time. Alpha is smaller for DOU than TOU, and this could be due to there is kind of a bias approach in the Berg method where you have to determine the surface water interface. So, and since the micro profiles were done in 100 micron increments, it's actually quite large for determining the surface water interface. So this would be easier done if they were smaller increments, but at that point in the sensor capability, the best way to do it is 100 microns. So at that point, it's just the way it has to be done. And it could also be smaller because that there could also be another process happening within the sediment, which is benthic fauna-mediated O2 uptake, which could be being done by bioturbating organisms. And then also it is determined from all of this that we are going to stick with the average TOU parameters, except for the middle site, to do the benthic estimates because they are most reliant, and we're looking at it as a whole benthic compartment contributing to the overall production. So we're assuming that this is a better way to do it because that's also what has been being done in literature in the past. Future work for this will include measuring the chlorophyll A samples that were collected during the time. So then we can look into basically the cell-specific chlorophyll A to see if there's a higher chlorophyll A present in the cells there, and that is why there's higher productivity and less productivity in the middle, rather than just biomass present. Then from here, the PE relationships will be used to combine with the measurements of the depth attenuation, surface irradiance to calculate the benthic primary productivity. And that is the main goal for the end of the study. I'd like to thank my advisor, Christopher Elgar, my committee member and kind of co-advisor from the Darley Marine Center, Jeremy Rich, his research assistant, Sean O'Neill, who helped me a lot, and my other committee member, Katia Fennell. Thank you.
Thanks, great talk. Now, are there any questions? Oh, Dave Greenberg has raised his hand already, so go ahead, Dave.
Yes, okay, you hear me okay?
Yeah.
Okay, so question, just wondering about the variability in light penetration, whether that's what sort of timescales you're measuring it in and how quickly it might change, whether it changes every time a sea-doo goes by, or you know, with a cloud going over, or the changing tide, whether you've resolved that, and what you might see in that sort of variation.
Yeah, there actually is quite a bit of variation, even like you said with the cloud coverage. When the water column like light profiles were done, we did them, it's an estimate, so like it's only going to be as good as you can get it. And basically we tried to make it that I guess you're getting like the fullest light you can. So we made sure that when we were doing those profiles that we weren't getting any clouds over. So we were trying to do clear sunny days. So yes, there would be lots of variation, but I mean, it's really hard to account for that when you're trying to make a in situ estimate based on ex situ parameters determined.
Very good, thank you.
Anyone else out there with a burning question? No, doesn't look like it. Okay, well, thanks a lot, Emilee, and thanks to everyone for attending all the talks. Like I said, it's going to be a few weeks break before we start off again in the 2020-2021 season. So enjoy your Tuesday lunches, and we'll see you again soon.